Now we're going to be discussing normalization approaches. So why normalize? Imagine you sequenced two stool samples. Sample A gave 50,000 reads and sample B gave 500,000 reads. So both are from equally diverse cuts. If you use raw counts, sample B will always look richer simply because of deeper sequencing, not biology. Now total sum scaling or relative abundance. So it converts counts to proportions. For example, sample A has E. coli 5,000 upon 50,000 10%, while sample B would be 50,000 upon 500,000 10%. After TSS, we see that both the samples show the same abundance, making them comparable. Now the limitation of this TSS total sum scaling or relative abundance tool is that because proportions sum to 1, if E. coli increases, other taxa must decrease even if their absolute abundance stay constant. This is the compositionality problem, right? Now talking about CLR, which is centered log ratio. Formula is CLR of xi is equal to log of xi upon g, where g is the geometric mean of the counts in the sample. For example, sample with counts 10, 20 and 30. The geometric mean would be 10 times 20 times 30 upon when we multiply, and the whole 1 by 3. Around it comes out to be around 18.2. In the CLR, we calculate these are the values, right? So log of 10 upon 18.2, log of 20 upon 18.2, and log of 30 upon 18.2. We get these values. Now the data are in Euclidean space, suitable for proper distances. So CLR is the one that solves compositional bias, but the zeros must be replaced. For example, add 1. Now alpha diversity, which is within sample or intra-sample diversity. So firstly, we have the metric richness S, which denotes how many taxa are present. Example, sample A has 5 species, sample B has 20 species, but this ignores the abundances. That is, 19 rare species could make B look richer even if dominated by 1. Then we have Shannon index. H is equal to minus summation pi log pi, which is the entropy as well. Example, sample A is 50% A, 50% B, so H is equal to minus summation of these two components, that is minus of 0.5 log 0.5 plus 0.5 log 5 log 0.5, which gives this value. And sample B gives basically, if we have 90% A, 10% B, we have a smaller value here. Interpretation is that both samples have two species, but Shannon shows A is more diverse because abundances are even, right? Half of A and half of B species. In B case, we have a lower Shannon index value, which means that according to Shannon index, we have a lesser diverse sample because the abundances are unequally distributed, 90% of A and 10% of B. So that is one thing. Now PLU even as J is equal to H upon log S. So using the above example, we do H is given to us as this, S is 2, which is the number of samples. And when we divide, J is equal to 1 and J is equal to 0.47 for sample B. So J is equal to 1 means that it is perfectly even, and J is equal to 0.47 gives us the estimate that it is uneven. And which is also true because sample A has 50% A and 50% B, so it should be perfectly even. That is why the answer also complements it, and we have 1. Sample B is uneven, 90% A, 10% B, so that is why the value of J is less than 1. That is the PLU evenness metric, right? So it isolates the balance dimension. Now we have the Simpson index, so probability that two random individuals belong to same species. For example, sample A is 50-50, the one we discussed. Simpson gives us 0.5 as the answer, and sample B is 9. Simpson value as 0.82, 90% A and 10% B, which means that higher dominance is of this one. So interpretation is that sample B is dominated by one species reflected in a higher Simpson value, right? So Simpson index, the purpose is to give us the probability of two random individuals belonging to the same species. Moving on, rarefaction curves. So example scenario, we have soil sample hyperdiverse, curve rises steadily even at 1 million reads. Then we have vaginal sample, which is low diversity, curve plateaus at 10k reads. So takeaway is that for soil, you must sequence very deeply to capture rare taxa. For the vaginal microbiome, shallow sequences is enough to cover more diversity. Now the rarefaction protects against false claims like soil has more species than vaginal microbiome, when the difference might just be sequencing effort. Now talking about beta diversity between sample differences or inter-sample differences. So we have Jacquard, that is presence or absence. So the Jacquard formula, let me show you again. That is J is equal to 1 minus A intersection B, the number of elements present in this set of species, and A union B, right? So this is the formula. Sample A has ABC, sample B has ACDE. Shared, or basically the intersection, is AC. So the 1 minus AC is the number, and we take the number that is 2 elements, and union is ABCDE. So that is total number of elements in that, 1 minus 2 by 5. That is the same thing, and that becomes 3 by 5. So the answer is 0.6. So it is quite different. Interpretation is that it ignores abundance, only cares about overlap in species list. Then Bray-Curtis is something like we have the sample A and sample B. Shared is minimum of 10 and 5, that is these two compared, and minimum of 20 and 25, the same species compared in both the samples. We add these from basically getting the minimum and adding these, and also get the totals. Then we basically apply the formula 1 minus 2 times the shared abundance upon the total abundance, right? Shared abundance upon the total abundance, and there's a factor of 2 as well. So 1 minus 2 times of shared abundance, that is 25, upon the total abundance, 60. So 0.17 is the value that we are getting. That is quite similar. So both samples dominated by the same taxa, more similar than Jacquard suggested. Then we have the CLR. So for CLR, we just get the CLR values, that is the sample upon the geometric mean of those samples, and Euclidean distance we calculate, so it captures the proportional changes rather than the raw counts. Euclidean distance we'll calculate between this and these 2 points basically, right? So minus 1.1 plus zero point 0.7 whole square plus 1.1 minus 0 0.7 whole square and the square root. Then we have the rank based. We won't discuss this. Unifrac also we have. So imagine sample A is firmicutes and bacteroidetes. Sample B is an actinobacteria plus proteobacteria. Taxa sets share no deep branches. That is unweighted unifrac is almost equal to 1. Very different. Weighted unifrac if firmicutes are 90% in A and only 5% in B. Distance will also be high but less extreme if some deep branches are shared. Strength, it detects differences invisible to taxonomic resolution. Species may differ but be close relatives. Right, so now some examples. Same species with different abundances. Bray, Curtis, low distance, Jacquard gives us 0. If we have different species but same ranks, rank based distance is low and Jacquard is high. Taxa differ, so basically different species but same ranks, Jacquard is high. Taxa differ but are close on phylogeny, Jacquard high, Unifrac low. Taxa differ and phylogenetically distant, both Jacquard and Unifrac are high. Right, so when we have difference is high, that means the intersection between the species is low, Jacquard would be high. You can see that we're differing, so Jacquard is high. Taxa differ and phylogenetically distant, Jacquard is high. But if we have same species, that is the intersection between sets A and B is actually the same. A and B are overlapping. That is, we have same species. The Jacquard would be 0. So as you keep bringing the sets that we have further apart, right? Let me show you using this diagram. So for example, we have this initially A and B, right? So the intersection is high. So that means the Jacquard is closer to 0. As we keep bringing these outward, that means A and B have lower intersection now. That is, the number of species are differing more in the taxa, right? So the Jacquard value also subsequently increases. And if we have completely, mutually exclusive sets, that means that there are no species common to these, right? That means the Jacquard value would be equal to 1. So that's that. And